Hello children, and today we will be learning to dissect the given flower, to study display, to study and display the different whorls, dissect anther and the ovary to show the number of locules or the chambers. The material required for this will be hibiscus flower, the very beautiful flower, forceps, blade, slide, cover slip, needle, the A4 size paper for displaying the dissected part of the flower, and the dissecting microscope. Children, before we start the dissection, we have to divide the A4 size paper into one part which will allow us to display the apicalyx, another calyx, corolla, then monoadelphous stamens, polycarpally, syncarpus, gynoecium. So, and here we will be able to draw the chambers or the ovary. This is the hibiscus flower, which is also known as shoe flower in common language or China rose. Children, you can see the green part of the hibiscus flower. This part is called as apicalyx. This we have to dissect it with the help of the forceps. Children, I will be plucking one by one. You will be provided the same flower in the exam. One thing I always like to do is I take the one gum and put it on the paper so that I can stick these apicalyx on the paper and they do not dislocalize. For display it becomes very easy. There are 5 to 7 apicalyx, should be arranged in a circle. Just press it so they remain stuck to the paper. They will not be dislocalized. There are 5 to 7 apicalyx. Now this portion is the calyx portion, which is fused, and we have to remove with the help of the forceps. And keep it in the place where it is written calyx. You can see here 5 calyx are there, children, which are fused. 1, 2, 3, 4 and 5. They all are fused and they make a cup-like arrangement. And just keep it here. Then you can see 5 petals, which are also called as corolla. Pluck them out and arrange them in circle. Beautifully arrange them on the piece of the paper. This is how display should be done for the corolla. Then we have Children, this is the male whorl. Outside you can see, and there is a stigma attached to the style inside. Children, you can see there are so many huge number of anthers, kidney shaped anthers are there, which are fused on one single tube. Now only you saw it is dissected and divided into two parts. All these are kidney shaped anthers which are fused on one single tube. Since these are fused on one single tube, this is the single tube, and therefore it is known as monoadelphous. Now we will be dissecting it so that we can visualize the style with the help of needle. This is the style, children. Children, this is ovary and this is the style and these are stigma. You can see very beautiful stigmas. 5 stigmas are there. 1, 2, 3, 4, 5. This together, stigma, style and ovary together is known as pistil. There are 5 stigmas which you can see, which contains sticky material so as to pollen grain can stick on it and can germinate, and this sticky material provide the nutrition for the pollen grains to germinate. Then there is a tube, long tube, which you can see, and this long tube till the ovary is known as style, and here you can see the ovary, chambers of the ovary inside. Inside there is ovary. Now we will be dissecting the ovary and observing it under the dissecting microscope, the chambers of the ovary. This is stigma, style and ovary. Together stigma, style and ovary is known as pistil. Means I can say this entire portion is called as pistil. With the help of the blade, we will be taking the transverse section of the ovary so as to visualize the locules and the ovules, and keep it on the slide. Keep it on the slide. And observe it under the dissecting microscope. There are chambers. Observe it under the microscope and see there are chambers in the ovary, and there are, those chambers are known as locules. The chambers which are there, they are known as locules, and in the locules there are axially present ovules, and it shows axial placenta, and there are ovules in each chamber. In each chamber there are 2 ovules. There are 5 chambers, and those chambers are known as locules. 1, 2, 3, 4 and 5. 5 chambers are there. In each 5 chambers there are 2 ovules. Now you will have to draw the diagram of this transverse section of ovary. You will have to draw the diagram, children, for the transverse section of the ovary, and there are 5 chambers as you saw after dissection, and each 5 chambers there are 2 ovules. These are known as locules, and these are known as ovules, and this is the axis in the middle where all the ovules are attached. Axis. It shows axial placenta, and this is the way children, this is the way you will be displaying all the floral whorls of your hibiscus flower. Children, this is the way you will be displaying all the floral whorls of your hibiscus flower. And now you will be writing the formula. After the dissection only you will be writing the formula of the flower. So you will be writing BR, and BR is bracteate. BR means bracteate. Bracteate flower means small leaf like structure. Okay, BR means small leaf like structure. Then you will be writing this symbol. This symbol means actinomorphic flower with the radial symmetry. Means if you cut in any symmetry, it will be giving you 2 equal half. Radial symmetry. Then flower is a bisexual, male and the female, female and the male. This is female sign, this sign is known as female sign, and this sign is known as male sign. So it is a bisexual flower. Then we use the term K for the calyx, and these calyx are fused. 5 calyx are there, you have seen 1, 2, 3, 4, 5. So K5, which are all fused K5, and therefore we use bracket. Then we have C5. Here we are not using bracket because all the corolla are free, they are not fused with one another, therefore we will not write the bracket. Then A, anthers are fused and they are infinite in number. Children, you have seen here the anthers, the stamens which are there, they are so many in number, and they are all fused on one tube. They are present on one single tube, and therefore we call it as infinite number of androeciums are there, and therefore which are huge in number stamens, and which are fused on one single tube. This condition is known as monoadelphous stamen. This fusing of all the stamens on one tube is known as monoadelphous stamen. Then we have G for gynoecium with 5 fused carpels, and here we have underline under G. This underline under G or below G represents superior ovary or hypogynous condition. This line which is under G is representing hypogynous condition. This is the way your complete display is done in dissection of flower. Now children, display of this floral whorl along with the diagram is of 2 marks. Dissection and display, and 1 mark is for drawing the diagram of the ovary, and this completes your 3-mark dissection of flower in question number 2. Children, anthers are bean shaped, that is kidney shaped, and this is the stamen. These structures which you see here, this is known as anther, which contain pollen grains in it, and this is the stamen. There are question answer which are given in your practical book. So the first question is, what is a flower? The flower is a seed bearing part of the plant which consists of reproductive organ, that is stamen and carpels, that are typically surrounded by a brightly colored corolla, which are known as petals, and a green calyx sepal. So you will have to write down these questions answer in your practical book. Second question says, what is the function of flower? The function of flower is to carry out sexual reproduction in plants through the formation of male and female gametes. Flowers ensures that the gamete fertilize to form the seed that further help in producing plants. Question number 3, which are the essential floral whorl and why? The flower whorls are calyx, corolla, and androecium and gynoecium. Androecium and gynoecium are called as floral whorls, essential reproductive whorls of the flower, because these are the structures that allow a plant to reproduce sexually. Since sexual reproduction inculcates evolution in species to ensure its continued survival, these whorls are very, very essential. So here you can see a picture of androecium and gynoecium. Androecium consists of anther and the stamen. This is stamen and this is the anther. Anther contain pollen grains on it. Together they are known as stamen. Then this is a male whorl, male essential whorl. Gynoecium consists of stigma style ovary, which together known as carpel, which is a female essential whorl. Corolla and calyx are the accessory whorls. Question number 4. What is the difference between polypetalous and gamopetalous corolla? Gamopetalous is the flower in which petals are fused or united and are known as gamopetalous flowers. Here you can see one picture of gamopetalous flower where the petals are fused. Polypetalous, the flower in which the petals are free, the petals are not fused. One of the example is rose, but here the picture is different, but you can identify. Even in rose the petals are not very much fused to one another. In your journal you don't have to draw this diagram, only to clear your doubt what is polypetalous and gamopetalous. I have added the picture in it. The fifth question, which are the different parts of the stamen and carpel? This is our stamen. Stamen is consisting of anther and the stalk, and the carpel consists of stigma, style, and the ovary. The last question is, how is brassica flower different from that of the hibiscus flower? Here there is a picture of the brassica flower. Children, you don't need to draw the picture, you only need to write the explanation, but for your knowledge I have uploaded the picture so that you understand clearly. Brassica is a genus of plants in the mustard family, that is Brassicaceae, and hibiscus is a genus of flowering plants in the Mallow family of Malvaceae. The genus is quite large, comprising several hundred species that are native to warm temperate subtropical and tropical regions throughout the world. So this complete our experiment of dissection of flower and displaying the floral whorls. Here I have to give you one meaning of the word, that is protrandus, the condition in which the male reproductive organ, that those are stamens of a flower, matures before the female ones, before the carpels are maturing, thereby ensuring the self-fertilization does not occur. Self-fertilization do not occur, and it will help in cross-pollination, cross-fertilization. The link is also provided below, and you can get the question answer uploaded in the link. Thank you for watching my video, and subscribe my channel if you have not subscribed, and share my videos. Thank you children, and like my video.